Good morning, students. Today we are having Immunology lecture number 8. Today we are going to talk about specific prevention and treatment of infectious diseases. We will talk about therapeutic, prophylactic, and diagnostic immunological preparations. In other words, we will talk about vaccines and sera that are used for prevention or prophylaxis, and sera that are used for treatment or diagnosis of infectious diseases. The first one that we should discuss is the definition of immunoprophylaxis and immunotherapy, and which immunobiological preparations are present at that moment. Immunoprophylaxis. It is a complex of public health measures directed on creation of artificially acquired immunity. Complex of health measures directed on creation of artificially acquired immunity. You remember there is a natural immunity and there is artificial immunity. So immunoprophylaxis is directed for creation of artificial immunity. It may be directed for creation of active specific immune response or active immunity that is mediated by vaccination. Vaccines are used to make active immunity because it causes production of antibodies. Immune system starts to work itself to produce antibodies. That's why it is active immune response. Or immunoprophylaxis may be used to create passive specific immunity that is mediated by injection with therapeutic sera or immunoglobulins. It is called passive immunity because the immune system doesn't need to produce antibodies itself. Antibodies or immunoglobulins are injected into human organism, so immune response is present, immune response is activated immediately. The aim of immunoprophylaxis is to provide specific protection of the human organism against infections. It is the main or important purpose or aim of immunoprophylaxis. Immunotherapy is treatment of humans with immune preparations to induce an adequate immune response against pathogens. So, immunoprophylaxis means protection against different infections. Immunotherapy is treatment. So, immunoprophylaxis is used when patient hasn't infected yet. Immunotherapy is used when patient is infected, was infected, when we should fight, when we should help him. Immunobiological preparations are classified into three big groups. These are vaccines, therapeutic serum, and immunomodulators. We will talk about them. But firstly, we should talk about history of immunization, how it developed, because we should know that immunization was difficult. Firstly, era of vaccination started from English doctor Edward Jenner. English physician Edward Jenner observed that milkmaids stricken with a viral disease called cowpox were rarely infected with the same disease, smallpox. So milkmaids, these are women who work with cows. So At that time, one viral disease was widespread. It was cowpox. So Edward Jenner noticed that this woman um, almost was not infected via smallpox, another disease, but the diseases are the same. So Jenner took fluid from a pastel of a woman who had cowpox and injected this fluid into a healthy people. And this manipulation was called variolation. Variolation. What it was? Inoculation with pus from a patient with a mild case of smallpox has been in use for over 1,000 years. So prevented people against smallpox by injection of fluid containing cowpox viruses. And in 1796, Jenner started on a course that would ease the suffering of people around the world for centuries. So, vaccination. We can't call it vaccination. It wasn't vaccination. It was variolation when he injected viruses from fluid into human. So the first one, this era of vaccination started from the prevention against the first disease, so that was smallpox. In this photo, we can see process of variolation. This man is Edward Jenner, and in this photo we can see he vaccinated James Phipps with cowpox virus that was present in fluid isolated from cows, and he created resistance in man in this boy and other people against smallpox. Then, after some years, another scientist, French scientist, Lois Pastor. You remember that Lois Pastor was the founder of microbiology. In France, in Paris, there is a famous scientific institute. Firstly, it was laboratory, then it is Institute of Louis Pasteur. They create vaccines and sera. They are working with different microorganisms. So, in 1879, Louis Pasteur showed that chicken cholera, weakened by growing it in the laboratory, could protect against infection with more virulent strains. It was very funny discovery in medicine. Louis Pasteur and his workers worked with chicken cholera. Chicken cholera can infect just chicken, but can't infect human. So they worked with the pathogen that caused chicken cholera. It was summer, and there were holidays. There were holidays, and they didn't work during 1 month. And his lab workers, so Louis Pasteur, forgot to save bacteria that cause chicken cholera in fridge. You know that if we want to stop multiplication of bacteria, we should put in a fridge. Low temperatures inhibit multiplication of bacteria. So lab workers in Louis Pasteur laboratory forgot to put this bacteria into the fridge. So bacteria grow in the tube in the laboratory, and they became weak. They became weaker than bacteria that, for example, are just isolated. And then Louis Pasteur showed he was very angry for these lab workers, and they continued to work with this bacteria. They injected this weakened bacteria into chicken, but nothing happened. Chicken didn't die because of cholera. They injected this weakened bacteria that they forgot in the laboratory once more. But cholera in chicken didn't develop once more. Then Louis Pasteur injected new virulent fresh strains. I mean that strains that they're isolated from other chicken. They injected new strains, new bacteria into animals. All chicken had to die, but nothing happened. So Louis Pasteur was a great scientist. He had a great brain, we can say. So he understood that the first injection of weakened bacteria produced resistance to virulent strains. That's why this experiment was happened occasionally, but it was the first case when Pasteur understood how they could create vaccines. And the first vaccine that was created in the world was made in 1881 by Louis Pasteur. He showed in a public experiment at Polysaccharide in France that his anthrax vaccine was efficient in protection of sheep, goat, and cows. So anti-anthrax vaccine was the first vaccine. He weakened Bacillus anthrax. I hope you remember from microbiology that anthrax is caused by Bacillus anthracis. Bacillus anthracis. This disease is transmitted from animals, cattle, goat, sheep. He weakened Bacillus anthracis by temperature, by high temperature. So anti-anthrax vaccine was the first one that was created in the world. After that, in 1885, Pasteur developed a vaccine against rabies based on a live attenuated virus. Anti-rabies vaccine was the first antiviral vaccine. Anthrax is bacterial disease, so anti-anthrax vaccine was the first antibacterial vaccine. Viruses, if you remember from history, in microbiology, that viruses, these microorganisms, were discovered just in 1905. In 1905 by Russian scientist Ivanovsky. But Pasteur made vaccine against rabies earlier. He discovered this vaccine, he created this vaccine earlier, in 1885. Earlier than discovery of viruses. He even couldn't see rabies virus because it was impossible. Viruses are very, very small. They are tiny microorganisms. It was impossible to see these microorganisms in microscope, under the microscope. So it was a great discovery that Pasteur created vaccine, even when he couldn't see microorganisms. After that, in 1886, Salmon and Smith developed a heat-killed cholera vaccine. Cholera was a widespread disease, so it was very important to create a vaccine against cholera. Over the next 20 years, killed typhoid and plague vaccine were developed. You also remember that plague, that is also called Black Death, killed a lot of people. And in 1927, BCG vaccine against tuberculosis was created. This vaccine is used today in all countries from all over the world. Today it is impossible to understand how people lived without any vaccination, because today we are protected. You and me and other people in all countries are protected against a great amount of microorganisms. Vaccination against infectious diseases starts from the first day of the birth of the newborn. And it is impossible to understand how people lived. They died. A lot of people died because due to tuberculosis, due to rabies, cholera, typhoid, plague killed a lot of people. But today we live, today we can live, today we are not afraid to live, and we even don't think about it, that every day we are protected by vaccines. In this photo you can see the first vaccination against rabies when Pasteur vaccinated Joseph Meister, this boy was bitten by an infected rabies dog. He almost died, but his mother brought this boy to Pasteur laboratory. He cried, he asked to help this boy, but Pasteur was very afraid to vaccinate this boy because anti-rabies vaccine had no experiments, wasn't used against vaccine in people. There were a lot of experiments with anti-rabies vaccine with dogs, with animals, but it was the first experiment with boy. So Joseph Meister could die in any case due to rabies without vaccination or due to vaccination, but they tried and it was successful. Joseph Meister was alive, and he worked as a security in Pasteur's Institute. What about vaccination today? Vaccines have been made for only 34 of more than 400 known pathogens that are harmful to man. So not all pathogens may be prevented, not all diseases may be prevented by using of vaccines. Just some of these. Immunization saves the life of 3 million children each year, but the 2 million more lives could be saved if existing vaccines were applied on a full scale worldwide. And the first immunobiological preparat that we will discuss is a vaccine. What is a vaccine? Vaccines are preparations of antigens whose administration artificially establishes a state of immunity without causing disease. So vaccines are preparations. These are antigens, killed or attenuated, whose administration establish immunity without causing disease. Vaccines must not cause disease. Sure, there are complications of vaccines. We will talk about it later. So vaccines are designed to stimulate the normal primary immune response. There are a lot of vaccines. They are classified into live vaccine, killed or inactivated vaccine. Live vaccine are also called attenuated vaccine because they contain attenuated live microorganisms. Killed or inactivated contain dead pathogens that are not living. Then polysaccharide vaccine, then conjugate vaccine, toxoids that contain not harmful toxins, and new generation of vaccines. These are recombinant vaccines or RNA vaccines. And the first one that we are going to talk, these are live attenuated vaccines. Live attenuated vaccines contain attenuated or virulent or non-virulent bacterial cells or viral particles. They are best at stimulating both a humoral and cell-mediated immune response. Frequently, only single introduction of live vaccine is enough to induce long-lived appropriate immunity. Most vaccine strains are good immunogens. It means that they cause good immune response. Good immune response means activation of T cells and B cells. Live vaccines are very dangerous for immunocompromised patients. Immunocompromised patients are especially HIV patients or patients with primary immune deficiency, because they carry a serious risk of causing other disease. For example, we vaccinate the patient by BCG vaccine. I hope you remember what is BCG vaccine. It's vaccine against tuberculosis. It is live vaccine. But in immunocompromised patients, this vaccine can cause tuberculosis. How to create this vaccine? So live attenuated vaccine contains attenuated microorganisms. How to make these microorganisms, virus and bacteria, weak? How to make them weak? The first way, it is screening of microorganisms which possess strongly reduced pathogenicity and high pronounced immunogenicity. The first one, when we look for microorganisms, look for bacteria that pathogenicity is weak, that pathogenicity is low. For example, BCG vaccine was created due to this way. BCG vaccine contains Mycobacterium bovis. You know that tuberculosis in men is caused by Mycobacterium tuberculosis, but it is a very pathogenic microorganism. Infective dose of Mycobacterium tuberculosis is very low. So they looked for and they found out that Mycobacterium bovis and Mycobacterium tuberculosis had the same antigens, but Mycobacterium bovis is less virulent. It's almost non-virulent for human, but virulent for cows, for cattle. So BCG vaccine was created with using of non-virulent or less virulent strain species of Mycobacterium. And another way of production of live attenuated vaccine is attenuation of microorganisms, attenuation of bacteria or viruses by chemical, physical, for example, high temperature factors, or by long cultivation in organisms of unsusceptible animals. Organism of unsusceptible animals, for example, when pathogen is harmful for human but non-pathogenic for animals. when we cultivate it in rabbits, for example. In rabbits, it lost pathogenicity, microorganisms lost pathogenicity, and maybe this strain may be used in creation of vaccine. What are these vaccines? What are these vaccines? Examples of live vaccines. It may be antibacterial vaccines or antiviral vaccines. What about antibacterial? Live vaccines are used against a great number of viral infections. This is polio vaccine. For example, Sabin vaccine that was produced by Sabin. There are two types of polio vaccine, Salk vaccine and Sabin vaccine. There were two doctors who worked at one time, and they created two different vaccines. Sabin created live polio vaccine, Salk created killed polio vaccine. Mmr vaccine. We will talk in microbiology. Mmr vaccine protects human against measles, mumps, and rubella. The first letters, measles, mumps, and rubella, means these diseases. Then vaccines against chickenpox is also live vaccine. Vaccine against hepatitis A, yellow fever, and other diseases, other viral diseases. Examples of live antibacterial vaccines includes BCG vaccine that is used against tuberculosis, or vaccine against typhoid fever. The another type of vaccine is killed or inactivated vaccine. How to kill microorganisms? How to kill microorganisms for creation of vaccine? The first one, it's inactivation or killing of microorganisms by exposure to denaturing agents. What are these agents? The first one is high temperature. High temperature may attenuate or may kill microorganisms. It depends on the temperature. Ultraviolet irradiation is also used to kill bacteria, and different chemicals, for example, formaldehyde. It's used to kill microorganisms. What about killed vaccines? It requires several doses of killed vaccine to achieve desired response. I have just said in live vaccines that frequently it needs just one dose of vaccination. Killed vaccine require several doses, and they predominantly produce humoral immunity, unlike live vaccine that produce both humoral and cellular immunity. But there is also advantages. There are positive moments in killed vaccines. They are safer. They are safer than live vaccine, but not so much effective than live vaccine. Killed vaccine may be used for vaccination of immunocompromised patients. There are also antiviral and antibacterial killed vaccine. For example, viral killed vaccine includes vaccine against poliovirus, poliomyelitis, Salk vaccine, against rabies, against COVID-19. For example, it is Covaxin. Covaxin is Indian anti-COVID vaccine, or Sinopharm. It's Chinese vaccine against COVID. It's also killed. Most bacterial vaccines are killed vaccines. Killed ones are typhoid, pertussis, and others. The third type of vaccines are toxoids. They are made from inactivated exotoxins from toxigenic bacteria. from toxigenic bacteria. What are these toxigenic bacteria? These are those bacteria that produce very harmful toxin, and the toxin mediates all the pathogenesis. I say to you a lot of times in microbiology that every clinical symptom, any clinical symptom that you can see in the patient, in most cases is mediated by action of toxin or enzymes, aggressive enzymes. So what are these toxigenic bacteria? Corynebacterium diphtheria that cause diphtheria, Clostridium tetani that cause tetanus, or Vibrio cholera that cause cholera. How to produce toxoids? So toxoid contains toxin, exotoxin. We should inactivate it. How to inactivate bacterial exotoxin? The first one, by treatment of the toxin with formalin at 37 degrees Celsius during 30 days. As a result, protein or toxin loses its poisonous but preserves its immunogenicity. It is not harmful for the human, but it causes immune response. It is immunogenic. It causes immune response, activation of immune cells. Toxoid induces antitoxoid antibodies, which are also capable of neutralizing the toxin. So after injection of toxoid, immune system produce antitoxic antibodies that circulate in the bloodstream, and they neutralize toxin that is produced by toxigenic bacteria when they enter human organism. What are examples of toxoid vaccine? These are vaccine against diphtheria. You remember, we studied it in microbiology. DTP vaccine, for example, diphtheria, tetanus, and pertussis. DTAP, TDAP, they are the same. In different countries, they are called. Then vaccine against tetanus, there are vaccine toxoid just against tetanus, and cholera vaccine. The another type of vaccine is polysaccharide vaccine. Polysaccharide vaccine consists of capsular polysaccharide of some bacteria. Some bacteria like Streptococcus pneumonia, for example, we studied it previous semester, consists polysaccharide capsule that is very immunogenic. Polysaccharide capsule is very immunogenic, and we studied in immunology when we talked about immunogens. There are T-dependent antigens and T-independent antigens. So polysaccharide capsule of Streptococcus pneumonia is very immunogenic, and capsular antigen activate directly B cells without activation of T cells. So capsular polysaccharide vaccine produce only immunoglobulin M. IgM can circulate in human bloodstream during short period. It is short-lived immunoglobulin. This vaccination is used rare today. They have been replaced with conjugate vaccines. Conjugate vaccines, I will tell you later. What are conjugate vaccines? What are polysaccharide vaccines? They include vaccines against Streptococcus pneumonia, PPSV, pneumococcal polysaccharide streptococcal vaccine 23, because it comprised of 23 capsular serotypes of Streptococcus pneumonia. Streptococcus pneumonia may consist any variant of capsule. There are about 23 variants of capsular antigens there. So there are about 23 serological types of Streptococcus pneumonia that cause pneumonia in patients in any country. So this vaccine contains all 23 capsular antigens. Once more, these vaccines were replaced. They are almost not used today. For example, Pneumovax was used during long period. These vaccines are not able to activate T helper cells. That's why memory cells are rarely produced. And these vaccines were replaced with conjugate vaccine. Conjugate vaccine consists of capsular polysaccharide antigen conjugate to protein, usually toxoids that activate T-cell dependent immune response. I have just said that polysaccharide vaccine don't activate T cells. That's why this capsular polysaccharide antigen was conjugated or are conjugated to protein that activate T cells. That's why conjugate vaccine are able to activate both T cell and B cell immunity, and they cause production of both IgG and IgM. Conjugate vaccines include vaccines that are against Haemophilus influenza bacterium type B. This bacteria causes wide range infections, meningitis, otitis, pneumonia, especially in children. And vaccine against Streptococcus pneumonia, that is pneumococcal conjugate vaccine PCV13. You can see it here. Vaccine that is conjugated in this vaccine. Bacterial capsular antigen is conjugated with diphtheria toxin. With diphtheria toxin, it is protein, so it activates both cellular and humoral immunity. Vaccines against Neisseria meningitis also are conjugated. The another type of vaccine is recombinant vaccine. Recombinant vaccine may be classified into recombinant antigen vaccine and recombinant vector vaccine. Firstly, recombinant antigen vaccines were created. Was the first vaccines against all recombinant vaccines. What about this vaccine? How are they created? What do they include? For creation of this vaccine, firstly, we should say that for creation of this vaccine, there was studied which antigen is responsible for pathogenesis of disease. The first vaccine that was created, the first recombinant vaccine, was vaccine against hepatitis B. The genes encoding any immunogenic protein can be cloned in bacteria or yeasts by recombinant DNA technology. I hope you know that any gene in our organism or bacterial or viruses, any gene is responsible for expression or for production of any protein. So, for example, in hepatitis B vaccine, gene encoding for the HBs antigen is inserted into yeast cells. We should say that in hepatitis B, we will study it in microbiology. It's a hepatitis B virus, and it carries HBs antigen, a specific protein on its surface that mediate all the pathogenesis and disruption of liver cells, of hepatocytes. So for the vaccine, we don't need to take whole viral particle. It's very harmful. It's dangerous. We need just to take surface HBs antigen. How to do it? How to isolate protein from viral particle? We may isolate gene that encodes this antigen, that encodes this protein. So we isolate this gene and insert it into yeast cell. Yeasts are non-dangerous for human, and then yeast cells releases this yeast cell gets new gene, so it releases this molecule HBs protein into the culture media. Then this molecule is purified and used as the immunogenic vaccine. So vaccine against hepatitis B contains just HBs antigen, just the protein. It doesn't contain viruses, so it never causes hepatitis B. This vaccine is impossible. The using of this vaccine is impossible to cause disease because it doesn't contain any viral particle. Another example of recombinant vaccine are recombinant vector vaccines. These vaccines are the most used today, the most widespread today, because most famous vaccines against coronavirus are recombinant vector vaccines. What are they? They use a viral vector. It is attenuated virus that can't cause disease in human. This viral vector is used to deliver genetic material coding for a desired antigen into the recipient host cells. The attenuated virus is just a vector that doesn't cause infection but expresses the gene product. What are these vector microorganisms? They include vaccinia virus, canary pox virus. Canary pox virus causes disease in canaries, in birds. Canary, I hope you know what are these birds, but never causes infection in human. Attenuated poliovirus or adenovirus, special strains that never causes infection in human. So what about coronavirus? Coronavirus, you know, consists as protein that is responsible for adherence and destruction of lung cells. We don't need whole coronavirus, whole viral particle in this vaccine. We need just this as protein. How to isolate this protein from viral particle? We can isolate gene that encodes this desired antigen, S antigen in coronavirus, and inject this gene into viral vector, viral microorganisms that are non-dangerous for human. These vector viruses enter human organism and they have this antigen, S antigen. So immune system responds to this antigen by production of antibodies and activation of T cells against S antigen and protects human organism against coronavirus. What are these vaccines? These are, I have just said, vaccines against COVID-19, Sputnik V, it's Russian vaccine, Oxford AstraZeneca, that is British vaccine, and Covishield, that is Indian vaccine. Another example of recombinant vector vaccine is a vaccine against Ebola virus. It's Ervebo. It was created when epidemic of Ebola was in Africa in 2014. Another example of new generation vaccine, it's RNA vaccine. RNA vaccines contains RNA which acts as messenger RNA in introduction in host cells. Host cells build the foreign viral protein encoded in messenger RNA and stimulate an adaptive immune response to this protein. So RNA vaccine also don't contain any viruses, any viral particle. They contain just RNA with genes that are encoded, any important viral protein. So when they enter the human organism, they stimulate immune response to this protein, like S protein, for example. Examples of RNA vaccines are also vaccines against COVID-19. These are Pfizer BioNTech, that is widespread for prevention of coronavirus, or Moderna. Moderna is a pharmaceutical factory that works only with that creates only RNA vaccines, because its name, we can understand this from the name, RNA. So I have just said about complications of vaccines, about classification of vaccines, and I have said that vaccines is very important to prevent infectious diseases. But we should remember that any vaccine may cause complications after its using. What are these complications and what are contraindications to immunization? Who must not be immunized? Firstly, about vaccine prophylaxis. Vaccine prophylaxis may be used by two types. The first one, when we use vaccines, it's planned vaccination. Planned vaccination is a primary immunization that may be given to all children. At the age of 2-3 months, vaccination against hepatitis B, diphtheria, pertussis or whooping cough, tetanus, and polio, with use of DTP vaccine and polio vaccine. In all countries, children must be vaccinated against hepatitis B, diphtheria, whooping cough, tetanus, and polio during first 2-3 months after the birth. Then, in age of 13-15 months, in age about 1 year, these children must be vaccinated against measles, mumps, and rubella. It is in all countries. It is planned vaccination. Another vaccination is called vaccination in the case of epidemiological reasons. It happens when any infection happens in the region or in country. The fine example of epidemiological vaccination is a vaccination against Ebola and coronavirus. You remember that they started vaccination against coronavirus, against COVID-19, when pandemic started. Or Ebola vaccination against Ebola is made just in those African countries where it is present when epidemic is present. So vaccination in the case of epidemiological reasons is made in definite regions where some endemic infections could occur. In the case of the professional contacts with infectious agents, I've told you in microbiology, for example, vaccination against anthrax is used for risk group people who works with animals. Or in the case of danger of epidemics of infectious diseases, epidemics of flu or bite of suspicious animal. For example, in Russia, every year in October and November, they start vaccination against influenza because December, January, February are the months that are characterized by widespreading of influenza. So these vaccinations in endemic infections like for Ebola vaccination for risk group people who may be infected, for example, professional risk, vet doctors or people or farmers who work with animals, and vaccination um, in case of danger of epidemics. These are vaccination in the case of epidemiological reasons. Planet immunization. Planet immunization may be done by two vaccinations. The first one is primary vaccination. Primary vaccination when we inject just one dose of vaccine, like BCG, measles, mumps, and rubella, or yellow fever. But there are vaccines that need multiple dose vaccination: polio, DTP, hepatitis B. One dose of vaccines: BCG, measles, mumps, rubella, yellow fever, multiple dose vaccines, polio, DTP, hepatitis B. Coronavirus, COVID-19 is also requires multiple dose vaccines. You know that it's necessary injection of two doses to be prevented against coronavirus. But there is no vaccine when just single injection protects human for all the life. No, it is impossible. Any vaccine protects the human just for some years, no more than 7 years, for example. Any vaccine requires booster vaccination. Once more, there are vaccines that may be injected just singly, like BCG. It is injected after the birth, right after the birth, or multiple dose. But both vaccines that requires one dose vaccination or multiple dose vaccinations requires booster vaccination. It is used to maintain immunity level after it declines after some time, after some years. DTP, measles, mumps, rubella, BCG, and others. You know that, for example, COVID-19 vaccine protects just for 1 year. So any vaccine requires booster vaccination after some years. Some vaccines requires it after 1 year, some vaccines after 5 or 6 years, like BCG. There are some contraindications to vaccination. Not all people can be vaccinated. So who must not be vaccinated? These are allergic patients. If patient has fever, you understand that it is impossible. We must not vaccinate such people. People with HIV infection. People with immunodeficiency, especially primary immunodeficiency. Patients after immunoglobulin administration. You understand, if immunoglobulin are injected into the patient, for example, against any disease, we must not vaccinate this patient against this disease, because antibodies, immunoglobulins, destroy antigens in vaccine, and vaccine will be not effective. Immune response will not activate. Patients with neurological disorders are also not vaccinated. Children who are birth premature birth people, children. Patients who had reactions to previous vaccine are also not vaccinated. Patients with simultaneous administration of vaccines, and patients with thrombocytopenia. Such people must not be vaccinated at all, or for example they may be vaccinated after some time. I mean, for example, if patient has fever now, we may vaccinate them, this patient after some time when fever will finish. Or if patient has a secondary immune deficiency, we may treat secondary immune deficiency, then we may vaccinate this person. What are complications of vaccines? What may happen if we vaccinate the patient who must not be vaccinated? What are these complications? The first one, the cross-reactive antigens of vaccine strain could cause autoimmune disease. Cross-reactive antigens that react antigens in vaccine may react with each other, and they may cause autoimmune disease when immune system recognizes our cells as foreign and destroy it. These are autoimmune diseases like rheumatic fever, for example. Some vaccines, for example against rabies, could cause encephalitis. The third one, vaccines can cause other pathological states. In the case when patient has acute cardiovascular disease or diseases of respiratory system, kidney, pathologies of nervous system, and other chronic disease or immune deficiency. When patient has diseases of the systems, pathologies of the systems, vaccination may cause disease, may cause pathology in this system. Vaccines also can cause strong allergic reactions. The list of these diseases is specific for every vaccine. You may take a description for any vaccine and read there what are complications. The first one is allergy. It may be like anaphylaxis, for example. Anaphylaxis is the most frequent complication of vaccination. What else? The danger of overload of the organism of children by alien antigens, especially during the first year when child gets from 4 up to 5 vaccination, that can cause pronounced sensitization and appearance of heteroallergies. During the first year of the life, the child gets a lot of vaccines. The great amount of vaccines, more than other years. But you know that the first year immune system is formed, it develops. So at that period, it can cause pronounced sensitization and appearance of allergy. What about immunization? Immunization in your countries. Sure, it is impossible. There are a lot of students from different countries. It's impossible to show you a list of vaccination in all countries. I will show for India and for Egypt. Remember, please, that in all countries, there are the same vaccines that are necessary, that are obligatory for using for all the children. And you may work in any country and you will understand that they are the same. So there are some vaccines that are different for these countries. We will talk about it. For example, in India, what are vaccines that are necessary for any children? The first one is BCG vaccine, that is given in birth, and hepatitis B vaccine that also given in birth. Then oral polio vaccine against polio that is given in birth, then in 6 weeks, 10 weeks, 14 weeks, and so on. Then injectable polio vaccine, maybe oral polio vaccine or injectable polio vaccine. Then vaccine that includes DTP, hepatitis, Haemophilus influenza type B and hepatitis B. It is a pentavaccine that consists that prevents against 5 diseases: diphtheria, tetanus, pertussis, Haemophilus influenza, and hepatitis B. Then or DTP, then measles, mumps, rubella. So these vaccines are given for all children against tuberculosis, hepatitis B, polio, diphtheria, tetanus, pertussis, hepatitis B, Haemophilus influenza, or only diphtheria, tetanus, pertussis, measles, mumps, and rubella. And there are vaccines that are injected just in endemic districts in India. For example, vaccine against Japan encephalitis. Then pneumococcal conjugate vaccine, and vaccine against rotavirus. It's a virus that causes severe enteric infection in children. I hope you understand vitamin A is not vaccine, it's just in this list. And tetanus toxoid is also its booster vaccination of DTP, especially for pregnant women. What about Egypt? Vaccines, once more, vaccines that are used there are the same. Most vaccines are similar. What are they? The first one, it's vaccine against polio, oral polio vaccine or Sabin's vaccine that is used for prevention of poliomyelitis. DTP or DPT, diphtheria, pertussis, tetanus. Then hepatitis B vaccine. Vitamin A, it's not vaccine. Booster vaccination against polio. Measles, mumps, rubella in age of 1 year. Oral polio vaccine. Diphtheria, pertussis, tetanus, it's a booster vaccination. And meningococcal vaccine. So vaccines are the same. Vaccine against polio, tuberculosis, diphtheria, tetanus, whooping cough, hepatitis B, measles, mumps, rubella, measles, mumps, rubella, and meningitis, Neisseria meningitis. For example, we can compare Egyptian and Indian immunization schedule. They are distinguished just in one vaccine. I'm talking about obligatory that are used for all. For example, in Egypt, they use meningococcal vaccine. Today we also use in Russia vaccine against meningitis. It's also necessary. So we have just said about vaccines that contain antigens. Now let's talk about immunological preparations that contain antibodies. So these preparations are used for passive immunization, while vaccines are used for active immunization. Passive immunity can be used to prevent diseases when there is no sufficient time to develop an acquired immune response through vaccination. Actually, if we inject vaccine, it takes about some time, several months, to produce immunity, 2 months, for example. But if we have no this time, we should inject serum. We should inject immunoglobulin, for example. But the passive immunity obtained by this method is limited to a few weeks. Passive immunity is active just during short periods of some weeks, while active immunity that is provided by vaccines active for some years. Passive immunization is provided by injection of serum, pooled gamma globulins that contains various antibodies, specific immunoglobulins or specific antitoxins that provide immediate protection. Serum, pooled gamma globulins, specific immunoglobulins or specific antitoxins provide immediate protection. Protection against disease is formed immediately, right at the moment of its injection, moment of this administration. Antitoxins are used to neutralize the toxin. So antitoxin consists antitoxic antibodies. Antitoxic sera are employed for treatment and for prophylaxis of tetanus, gas, gangrene, and botulism. We have said about it in microbiology. Antimicrobial sera are used against anthrax, encephalitis, and influenza in the form of globulins. So there are two types of sera that may contain antibodies against toxin and antibodies against bacteria or viruses. Antibodies against toxin are present in antitoxins. They neutralize toxin. Antibodies against bacterial viruses contain antibodies that destroy bacterial cells or viral particles. Sera that are used in medicine may be classified into some groups in accordance to its using. There are therapeutic serum. It is serum that's used for treatment of infectious disease by injection of antitoxin or serum containing specific antibodies. When patient is infected, we may administrate therapeutic serum. Prophylaxis serum is another one. It is injected into someone's blood to protect them against a poison or disease. For example, when other people are infected, we can prevent people who are not infected yet. And the last one is diagnostic serum. That's serum that's used for diagnosis of infectious diseases in serological tests. In immunology, this week you discussed this serum. These are commercial serum that contains known antibodies. For example, serum against E. coli, serum against Brucella. It contains antibodies against known bacteria. They are used in serological tests. Sera may be classified into two groups in accordance to its origin. These are heterologous serum and homologous serum. Heterologous serum is serum that's obtained from animals after immunization. Hetero means from other organisms. For example, from horses. Why do we use horses? Horses are big animals. So amount of serum is great in them. We may immunize horses and get serum with antibodies. Immunize means inject bacteria or virus. Immune system of horse produce antibodies, and we can collect serum from this horse and use it for treatment of people. Or homologous serum is serum that's obtained from human after disease. We always should remember that any foreign protein serum contains antibodies. Antibodies are protein. Any foreign protein may cause anaphylactic reaction. Antibodies that are injected for prevention of diseases may be classified into monoclonal antibodies and polyclonal antibodies. Monoclonal antibodies are antibodies produced by a single clone and directed against a single antigenic determinant. Antigenic determinant, I remind you, it is a part of antigen that is specific and that causes immune response. Any antigen contains antigenic determinant and carrier molecule. Carrier molecule is not specific, it is a larger part of antigen. It doesn't cause immune response, but antigenic determinant or epitope causes immune response. So monoclonal antibodies, they produce, these are antibodies that may be joined with a single antigenic determinant. Polyclonal antibodies are antibodies against various epitopes of the antigen. Various epitopes. What about injection? What about administration of vaccines and serum? Firstly, we can inject vaccines or serum via a great number of routes. For example, intradermally into skin. BCG vaccine is injected intradermally. That's why there is a scar after injection. If you look at your left shoulder right now, you can see a scar on left shoulder. It is after BCG vaccination because it is administered into the skin. And BCG vaccine is live vaccine, you remember. That's why it causes local infection process in the skin. Then vaccines may be injected subcutaneously under the skin. Most vaccines are injected intramuscularly, into muscle. These are widespread. Intravenously, when vaccine injected into the bloodstream, it is rare. There are vaccines that are injected into the mucosal cells lining the respiratory tract through inhalation. You know maybe that today the doctors works under the developing of vaccine against coronavirus that may be inhaled vaccine. Orally, vaccines that are administered orally into the gastrointestinal tract, for example, vaccine against polio. This is Sabin vaccine. Sabin vaccine against polio that are injected orally when live viruses from polioviruses interact with intestinal cells in small intestine, and they produce immunity. The effectiveness of vaccines depends on how they are introduced into the body. For example, in this table you can see a route of administration of vaccines that are used in all countries. The first one, BCG, it's used intradermally, injected intradermally. I have said about it in left arm. Then DTP only intramuscularly, oral polio vaccine orally, hepatitis B vaccine only intramuscularly, measles, mumps, rubella vaccine subcutaneously. Tetanus toxoid is also administered intramuscularly. You also can see that are administered in different injection sites. Today I have said to you that injection of vaccine and serum may be harmful. I've said about complications, that any vaccine and especially serum contain foreign protein. I hope you remember from lecture number 4 in immunology that any foreign protein may cause allergy, may cause hypersensitivity type 1. What is name of this type? It is anaphylactic. Hypersensitivity type 1 is anaphylactic. Develops due to injection of foreign protein, food allergy, you remember. Today we are talking about vaccines and serum. So it is important to provide successful injection of vaccine or serum without development of allergy. How to do it? There is a method for injection of serum that is used to avoid allergy. It's so-called Bezredka's method. Bezredka was a Russian doctor who suggested to use it. Bezredka's method is the method of desensitization, which is necessarily used to prevent anaphylactic reactions in injection of serum. So we should decrease sensitization of the human who gets injection of serum. How to do it? Firstly, about 0.1-0.3 milliliter of serum is injected subcutaneously. And then we should observe the patient's reaction. This small dose may cause activation of some immune cells, but it doesn't cause, a small dose doesn't cause anaphylactic allergy. After 1-2 hours, in the absence of anaphylactic reaction, the rest of the serum dose is injected. Sure, if we can see an anaphylactic reaction, what are signs of anaphylactic reaction? You remember, it is redness, swelling, bronchial spasm. If patient has allergy, we will see these clinical manifestations in the patient. So we must not inject a rest dose of serum if we can see signs of anaphylaxis. If we can't see, we can inject a rest dose of serum. And another group, the last group of immunological preparations, these are immunomodulators. We have said about vaccines, serum, now about immunomodulators. Immunomodulators are substances that stimulate or suppress immune system. They can be classified into two groups, immunostimulators and immunosuppressive drugs. What are immunostimulators? They stimulate immune system by increasing the activity of any of its components, interferon or interleukin or colony-stimulating factor. They are used for treatment of infectious diseases, tumors, immunodeficiency states, and other states when immune system needs a booster. So immunostimulators stimulate immune system. Unlike another group that is called immunosuppressive drugs. They inhibit activity of the immune system. They are used to prevent rejection of transplanted organs for treatment of autoimmune disease and allergy. You know that sometimes we need inhibit immune system. When? For example, after organ or tissue transplantation, because immune system is activated and any transplanted organ or tissue is antigen for human organism, for a recipient. So we should inhibit immune system, inhibit immune response for successful transplantation, for avoid of graft rejection. Or, for example, in autoimmune disease, you remember that autoimmune disease is caused by activation of immune system against our self cells. So when immune system recognizes our self cells like foreign. So we should inhibit immune system in this case. We should stop activation of immune response against our self cells. How to do it? By injection of, by using of immunosuppressive drugs. Allergy is also pathological state that is called abnormal immune response, when we should inhibit mast cells, for example, or basophils, which which immunosuppressive drugs are used. For example, these are glucocorticoids. They inhibit both cellular and humoral immunity, and they have anti-inflammatory action. Glucocorticoids includes prednisolone, cortisone. They may be also used in treatment of allergy. They inhibit release of allergic mediators, histamine and heparin. Then, cytostatics. These drugs inhibit cell division. And another group is the last one, drugs acting on immunophilins. So, that's all. Thanks for your attention. I remind you that next week you will have microbiology colloquium. So prepare for this colloquium. For next week I will explain you microbiology.